Michael, and good morning, everyone. Okay, so today we'll be learning about the translocation in phloem, right? So, candidates, you should be able to explain the concept of source and sink and phloem loading as well as unloading in translocation according to the pressure flow hypothesis. Okay, so now generally, okay, translocation in phloem means the transport of organic substances, okay, in vascular plants. Okay, so now let's go to the source and sink concept. Okay, you can refer to the diagram here, right? So this is the xylem vessel and this is the sieve tube element that is found in phloem, right? Okay, so sources are site. Okay, example is photosynthesizing leaf and tuber, okay, in potato, where organic substances such as sucrose or sugar are synthesized. Okay, and then apparently this sucrose or sugar, okay, after they are being synthesized, okay, by the leaf, apparently this sucrose or sugar will be loaded into the phloem. Right, you can see here, this is the leaf that synthesizes the foods or the organic substances, to be more precisely sucrose or sugar, okay, is being loaded into the phloem. You can see here, right? Now, sink is the site, okay, where the sucrose is unloaded from the phloem. So apparently, this sugar, okay, from the leaf or from the source loaded into the phloem, and from the phloem, the sugar, okay, will be unloaded to the sink. Now, the sink, okay, example of sink is like roots, shoot, epices, flowers, fruits, okay, or germinating part of a potato, okay, or any part of the plants that needs sugar, right, sucrose. Now, let's start at the beginning, at the source cell there. Okay, how does this process occurs? Now, at the source, sucrose is actively loaded into the sieve tube elements. Okay, they'll be actively loaded. So this is an active process. Okay, and this active process occurs by mass flow or bulk flow. So meaning to say the intake or the loading of sucrose into the sieve tubes occurs in high numbers of sucrose, okay, or high concentration of sucrose, not little by little. Okay, so now this will reduces the water potential inside the sieve tube elements. Yes, because you have many or influx or loading of sucrose into the sieve tube elements. So the water potential inside the sieve tube elements reduce, right? Next, water from the xylem, you can see from here, the water from the xylem will diffuse into the sieve tube elements by osmosis. What happens after we have many or high number of water molecules inside the sieve tubes? Okay, this will generate high hydrostatic pressure in the sieve tube elements. Then along these sieve tube elements, okay, the phloem sap will flow from higher hydrostatic pressure region to a lower hydrostatic pressure region. Okay, so apparently when the water from the vessel element mix with the sucrose, okay, from the source cell in the sieve tube, this mixture is known as the phloem sap. Okay, next, the sugar is then unloaded at the sink through the plasmodesmata. So at the sink cell will receive the sucrose. So that's it. Okay, So this is the source and the sink concept generally. Okay. Now you are going to learn about the hypothesis on the phloem translocation. Okay, so I already explained to you about the phloem translocation. Now let's go behind the hypothesis that explain this phloem translocation. Okay, so hypotheses on phloem translocation, they are mass flow hypothesis, cytoplasmic streaming hypothesis, electro osmosis hypothesis, and peristaltic wave hypothesis. So apparently these four hypotheses, okay, they combine, okay, and it explain to you the phloem translocation that I've explained earlier. Right now, so let's go into detail on each one of these four hypotheses. Okay, the mass flow or pressure flow hypothesis. Now, on this hypothesis, there are two parts, the principle and the application. Okay, now let's go to the principle first. Okay, you can refer to this diagram here. Okay, this mass flow hypothesis, okay, stating that this A is the source that has low or lower water potential. Okay, and the source, example of this source is the photosynthesizing mesophyll cell of a leaf. Okay, so the water will diffuse or enter, okay, this A by osmosis, right, because of the low water potential in the A. Now, the increase of hydrostatic pressure in A will force the sucrose solution in A, okay, to move across the connecting tube B, okay, and this connecting tube B is known as the phloem sieve tube. Okay. Next, the mass flow of sucrose solution, okay, from sap, okay, to the C, right, and the C is known as the sink, right, which is example is the roots or growing shoots, okay, and other parts of the plants that needs sucrose. So the phloem sap travels from A to B to C. Next, what happens at the C? The water will diffuses out from C. Okay, you can see here how the water diffuses out from the C and enters back to the A through D. This is D, and this D is the xylem vessel. So this mass flow continues. Okay, it continues until concentration of sugar solution in A and in C are equal. So in nature, equilibrium is not reached because source, okay, always synthesize solutes to be utilized at the sink. Okay, so this is the mass flow hypothesis, which was proposed by Ernst and Munch. Okay, so this is about the principle. Now let's take a look at the application. Okay, this application is very similar to the ones that I have explained earlier. Okay, about the phloem translocation in plants. Okay, so again, proposed by Ernst Munch. Sorry, earlier I said Ernst and Munch, right? So it was Ernst Munch. Okay. So fluid flows from a region of higher hydrostatic pressure into a region of lower hydrostatic pressure. Now, candidates, when you write your answer in the essay, okay, always starts your essay with introduction, with an introduction. And one of the introduction that you can mention for this particular question, if it asks you about the mass flow, okay, of phloem translocation, okay, based on this Munch hypothesis, you can always start as your introduction by writing proposed by Ernst Munch. Okay. Transpiration pulls water up the xylem vessel. Now, candidates, I want to stress here. Always remember, the pulling of water in the xylem vessel is because, okay, it is caused by the transpiration which is at the leaf. Okay, so because transpiration in the leaf will cause the loss of water from the leaf mesophyll, okay, as water vapor, and this water vapor must be replaced, and the water vapor is being replaced by the water from the xylem, and this will create a strong pull up of force, okay, in the xylem vessel. And then the transportation of phloem sap in the phloem is by mass flow and is an active process. Okay, sucrose is actively transported or loaded from the source cell into the sieve tubes. This causes the sieve tube to have lower water potential. So water will diffuse into the sieve tube from the xylem via the osmosis. Okay, so the sucrose solution of phloem sap then unloaded into the zinc cell. Water will diffuse back into the xylem vessel because transpiration pulls water up the xylem vessels again. The higher water potential in the zinc cell and the lower water potential in the source cell will create a pressure difference, okay, causing a continuous flow of sucrose solution from the source to the sink. Okay, so this is the application on the hypothesis proposed by Ernst Munch. Okay, now let's go to the cytoplasmic streaming hypothesis. Okay, you can refer to this diagram here. Okay, so this is the sieve tube and this is the companion cell. Now, in this hypothesis, okay, the phloem sap circulates in the sieve tube. You can see here. Okay, phloem sap. Okay, this is the phloem sap, which consists of the sucrose and the water. Okay, it circulates in the sieve tube and can cross pore, right, at sieve plates due to movement of cytoplasm around within the sieve tube. Protein tubules in the cytoplasm will assist the flow of phloem sap in both directions across the pores of the sieve plates. So I'm sure you know this is the sieve plates, right? And then I'm sure you know that in the cytoplasm we have many protein tubules. We have learned this in the first term. So this is about the cytoplasmic streaming hypothesis. It explains how this phloem sap travels from one sieve tube to another sieve tube through the sieve plates. Now, electro-osmosis hypothesis. Okay, you can refer to the diagram here. So, in this hypothesis, the movement of ions with solute solution, okay, in a negatively charged fixed porous electrical field. Okay. So sieve plate and phloem, they are the fixed porous negatively charged surface. Okay. This is the sieve plate. Okay, where's my cursor? This is sieve plate, and initially the sieve plate are negatively charged. This diagram here, it shows you the aftermath of it, where it became positively charged. But initially, it is negatively charged. All right. Now, how does it become negatively charged? Okay, because of the potassium ion, okay, being pumped into the sieve plates. okay, from the companion cell that diffuses into the other side of the sieve plate. So, okay, so meaning to say potassium pump, okay, between the sieve tube and this companion cell will pump potassium ions, okay, onto the sieve plate, right? Now, the potassium ion here, okay, will diffuse into the other side of the sieve plate. So this region here, okay, initially it is positively charged, and this region here will be negatively charged. Now, during mass flow, only the positive ions and potassium ions are able to pass through the sieve plates. Okay. Negative ions, together with water and sucrose, are repelled and accumulated above the sieve plates. When the concentration of the negative ions reach a certain critical value, okay, protons are actively pumped out from the companion cell into the sieve plate. This will decrease the pH, okay, pH in the sieve tube, and make the cytoplasm above the sieve plate as well as the sieve plate become positively charged. The accumulated negative ions, sucrose and water, okay, will cross the positively charged sieve plate. Since the sieve plate is positively charged, only the positive ions are repelled, and this causes the proton to be pumped back into the companion cell, and the cycle repeats. Okay, so this electroosmosis hypothesis explains how the sucrose, okay, which are negatively charged, are able to cross, okay, these sieve plates from one sieve tube to another sieve tube. Okay, now we continue with the peristaltic wave hypothesis. Okay, now let's continue with peristaltic wave hypothesis. Okay, now you can refer to this diagram here. Okay, now in this hypothesis, protein microtubules in the cytoplasm of sieve tube, you can see this one sieve tube, it has many protein microtubules. Okay, and it span, okay, these protein microtubules span across the pore of the sieve plates. You can see here, it span across the pore of the sieve plates and continues along the sieve tube. These protein microtubules can constrict and relax individually, okay, and it uses ATP in alternative pattern, which result in peristaltic waves. So these peristaltic waves will help to push the phloem sap inside the microtubules along the sieve tube, okay, from the source all the way to the sink. Okay, so that's about it. Okay, so hopefully you gain something here, and see you in the next class. Okay, so you'll be learning about neurons. Okay, bye.